How is systems biology used in the study of disease and in drug development? Let's remember that virtually all drugs are molecules that bind proteins and interfere with their molecular function. Here, for example, is a drug, the small green molecule, that binds a specific protein. In this case, the protein is phosphodiesterase. In the study of vascular diseases, it was found that phosphodiesterase breaks down a compound that causes vasodilation. Thus, by removing the compound, the protein causes vasoconstriction. The drug binds the phosphodiesterase and does not let it remove the compound, which leads, again, to vasodilation. This drug is, of course, the famous Viagra. The discovery and development of a new drug, therefore, is built on the following steps. First, selecting a target. Which protein should be attacked in order to fix the clinical problem? Target discovery requires deep molecular understanding of the disease. We need to know which proteins are involved in the disease and what will happen if we bind each of them. Of course, unlocking the molecular basis of the disease is crucial not only for drug discovery, but also for understanding the disease in general. Once we know which genes and proteins are involved in a disease, we can better predict how specific mutations in different genes will affect the patient, leading to better diagnosis and potentially for precision or even personalized care. Traditionally, this study hinges on the tedious work of molecular biologists who meticulously explore the function of each individual gene and protein. But this molecular approach is unlikely to give us the full picture anytime soon, or ever. We need the help of systems biology and its computational tools to sift through the results of high-throughput experiments that map the activity of thousands of genes, like the tool we mentioned in Unit 1. More importantly, we increasingly realize that proteins and genes act together. Interfering with the function of one protein may completely alter the way other genes and proteins act. This is the main cause of failure of new drugs. The drug may do to the target protein exactly what we wanted it to do, but the other proteins and genes in the system may respond to this change, leading to outcomes we cannot anticipate unless we consider the larger system, the network. On the flip side, the drug may also affect other functions of the target protein, affecting processes that we didn't consider and leading to severe side effects. Analyzing the networks of interactions between genes and proteins offer a powerful tool to address these problems. By knowing which protein interacts with which other proteins and which genes affect the expression of which other genes, we can get a better understanding of the molecular underpinnings of the disease. To identify a good drug target, one can look at these networks like military strategists look at a map, deciding where to attack. Network analysis may suggest that what we want to do in some cases is to attack from two different points with two different drugs, or with a drug that can bind two targets. This leads to the increasingly popular strategy of combination therapies and bispecific drugs. One vision of systems biology is the creation of dynamic models. These models describe the interaction between different genes proteins, and metabolites in a dynamic way. To determine whether a certain protein or a certain combination of proteins is a good drug target, we will perturb the dynamic model and see what happens to the other interactions in our model. Such models are developed today for specific biological pathways. But as more data become available, dynamic network models will be developed for whole cells or even for an entire organism. In principle, these models can also include the mutations that a given patient has and the drug or the drugs he or she are already taking. This will allow us to predict how a given patient will react to a given drug or to a combination of drugs, getting us closer to realizing the vision of precision medicine. Once we identify a putative target, we need to discover a molecule, which could be a small chemical compound like Viagra, or a large biological macromolecule like an antibody, or a recombinant protein. Traditionally, drug discovery is based on trial and error. Take a large bucket of molecules, throw them at the target you selected. If one of them binds, good. If not, bring another bucket of new molecules and try again until you find one that sticks. This obviously may take a long time. Advanced computational tools offer an alternative path. One of them is virtual screening, computationally predicting whether a given molecule will bind the protein in a way that will affect its function. Another computational approach is that of rational design, analyzing the structure of the target and designing on the computer a new molecule that will lead to the desired molecular effect on that protein. Combining the tools that systems biology offer for the selection of new targets with the tools computational biology offers for the design of new drugs is already leading to a revolution in the study of disease and in the preclinical development of new drugs.